Hello everyone, I'm Siham, a third-year PhD student at King's College London. I'm going to present our work taken to improve the detection of growth hormone-releasing hormone synthetic analogues for anti-doping purpose. Growth hormone-releasing hormone synthetic analogues are prohibited in sport in and out of competition under S2 category of the WADA Prohibited list. In this project, we are not looking at small molecules or small peptides, but we are looking at GHRH synthetic analogues that are greater than 3 kilodaltons. And WADA requires their detection in biological matrices, typically from 1 ml of urine at relatively small concentration of 1 nanogram per ml, which is the WADA minimum required performance levels. So GHRH are certainly available. Indeed, 2 synthetic analogues were identified in seized material by Norwegian and Belgian anti-doping laboratories. Cornell Laboratory developed an approach to detect intact GHRH synthetic analogues and 2 large metabolites in human plasma. Unfortunately, this approach requires immunoaffinity purification and nano LC-HRMS. This approach is difficult to run routinely. However, with all analytical means at our disposal, no adverse analytical finding has been claimed in testing figures so far. So we thought of looking at metabolites rather than parent peptides because the intact GHRH synthetic analogues won't be easily detected. As you might expect, GHRH synthetic analogues are very similar in structure to human GHRH. However, there are some chemical differences which can be key to what we want to look at. The included synthetic analogues in this project are tesamorelin, sermorelin, CJC-1295, CJC-1295 with drug affinity complex. Tesamorelin has the same amino acid sequence as human GHRH, but with modified N-terminus with 3-transhexenoic moiety. Sermorelin is a shorter fragment of GHRH with identical amino acid sequence. CJC-1295 is a modified version of the shortest fragment of GHRH with substitutions of 4 amino acids. D-alanine in position 2 instead of L-alanine, glutamine in position 8 instead of asparagine, L-alanine in position 15 instead of glycine, and leucine in position 27 instead of methionine. CJC-1295 with drug affinity complex, compared to the previous one, has an additional lysine in position 30, conjugated to maleimidopropionamide in C-terminal. As mentioned, we decided to look at metabolites. So we performed an in vitro study following the described protocol by the Belgian laboratory cited below, which is an accepted approach for in vitro drugs metabolism. After incubation of each peptides with human liver and kidney microsomes and serum, we were able to identify several metabolites via LC-HRMS. Far more metabolites were identified, but we actually selected the most significant metabolites based on the signal abundance in mass spectrometry and on their presence over a long incubation period. Selected metabolites are illustrated in figure 2, where I show the cleavage points colored markers are orientated toward the direction of the resulting putative metabolites, while red markers represent human liver and kidney microsomes in vitro model, and blue markers represent serum in vitro model. In the table on the right-hand side, I'm showing how many putative metabolites we obtained from just the 4 intact synthetic analogues. Apart from synthesizing the peptides, the whole work was very simple as illustrated in this slide. The process started from chemical synthesis of all the 19 selected metabolites by solid phase FMOC approach. Crude peptides were purified by preparative reverse phase HPLC. And because we wanted to be certain we had made the right materials, metabolites were characterized by HRMS. Then MRM transition were optimized. Last but not least, solid phase extraction was optimized for a better recovery. Then, spiked urine with metabolites at WADA MRPL or less was extracted by a simple solid phase extraction protocol prior to LC-HRMS analysis. This slide represents extracted chromatograms for tesamorelin M2 as an example. And what you can see, reducing concentrations from 10 nanogram to 1 nanogram per mil, which is the WADA MRPL. Then half MRPL down to 0.2 nanogram, where the chromatograms start to be a bit noisy, but still clearly shown. Our approach has accomplished in vitro metabolites identification, chemical synthesis of reference material, sample preparation optimization, and most importantly presented a pre-validated method with good detection sensitivity at lower than 1 nanogram per mil WADA MRPL. So I'm pleased to say at this point of the presentation that this is a simple and efficient analytical method ready to hand out for a routine validation and use in doping control. Sensitivity was good, easy method was built, and still need to be tested in suspect samples. But we are optimistic, and I hope by now you are optimistic too. With this, I have joined the end of my presentation, and thank you for your attention.